Alright, so we're going to look at differentiation for the phloem. So again, we're going to start with a meristematic cell, which is basically a cube with a nucleus. And then right away this will differ a little bit from xylem in that we're going to have a mitotic event, so that the mother cell becomes two daughter cells. And during that mitotic division, don't forget we're going to have lots of leftover connections through that cell plate, the plasmodesmata, and those will remain in place. And the plasmodesmata, you can think of it as a small strand of membrane with cytoplasm. We have our nuclei, so we're still kind of those cubes. There. And then we're going to start elongation through vacuolization. And pretty early on, there's going to be differences in the amount of elongation that occurs, so that the sieve tube member, or what will eventually become the sieve tube member, has much more elongation that occurs than in the companion cell. So here's that large central vacuole, the companion cell with its large central vacuole. And we still are highly connected through all of those plasmodesmata. We still have the nuclei, and then our large central vacuole. And then the sieve tube member basically becomes a zombie cell, and its nucleus disintegrates. It has these huge sieve pores that develop along the end. This is that sieve plate that develops. Now we have very little cytoplasm, just a kind of a thin strand running through that cell and then to the next. And we have basically all of this middle is going to be filled with photosynthate, which is things like sugars, sucrose, some amino acids. We might have a few mitochondria left over, but they're not acting like they would in a normal cell. We might have some membrane endoplasmic reticulum left in here, but really this is very basic what's going on in here. The companion cell is really what's controlling what happens within the sieve tube member. So it's kind of the controller of what you could probably consider a zombie cell now. It has no nucleus, so it's being completely controlled by the companion cell. Still have those plasmodesmata, and we'll see how those are used to load and unload photosynthate. And that is the mature form. They're still living. Both cells are still living, but the sieve tube member no longer has a nucleus. So we complete our Phloem differentiations, where we started with a meristematic cell, totipotent, basically a cube with a nucleus, undergo mitosis to form two daughter cells that then undergo elongation and vacuolization to eventually end with our two types of phloem cells, the sieve tube member with no nucleus, very little cytoplasm, and the companion cell with all of its complement parts controlling that sieve tube member.